Hello, everyone, and welcome to ACS Webinars, connecting you with the best and brightest minds in chemistry, live from Washington, D.C. I'm Michael David, and I'll be your host for today's webinar, which is being co-produced with the Science History Institute. My pleasure to introduce today's moderator, who is Cheng Fang, the Senior Vice President of Research and Development at Novus Bio, a clinical stage biotech. She is an accomplished neuroscientist with 2 decades of experience in neurodegenerative diseases with a broad scientific knowledge and hands-on experience. She has a successful track record of scientific publications and contributions. Dr. Fang also has a deep knowledge of drug development with both preclinical and clinical development experience. I would like to introduce Greg Sharples-Cook, Director of the Office of the President at the Science History Institute, who would like to make a brief announcement.
Good afternoon. The Science History Institute is a museum and library in Philadelphia that collects, preserves, interprets, and shares the stories behind the science. We are very grateful for the collaboration between ACS and our Joseph Priestley Society in producing this webinar. The Joseph Priestley Society promotes a deeper understanding of science, technology, and industry with an emphasis on innovation and entrepreneurship. Speakers are leaders from a wide variety of large and small chemical or life science companies and the financial, consulting, and academic communities.
Hello, everyone. Good morning, good afternoon, whatever time of day you are in. It's great to be here today, especially on World AIDS Day. Although our government has said we should no longer celebrate this day, we are. And I'm eager to go through some of the early history of the epidemic before I pass it over to Moupali Das, who will bring us very up to date on kind of where we are right now. So with that, where are we? Learning from AIDS. Very briefly, I'll go through a little bit of the early epidemic. I'll give a little bit of a San Francisco perspective, and again, take us up to some of the important developments that happened that brought us into the modern age. I'm going to start with a survey question. This always surprises me. And Michael, do you want to take this over and go through the audience survey?
Of course. How old were you in 1981?
It's a pretty easy question, Mike.
Pretty easy question. Just putting them through their paces. It looks like the younger end of the spectrum is a little bit more represented, with 34% being born after 1981. Next highest being 20 through 29, and after that, 30 and older. So that'll turn it back over to you, Paul.
It's thanks. It's strange to see 30 as old for somebody like me, but there you are. And I know for a lot of people, the AIDS epidemic seems like ancient history, but given the pandemic that we've just been through, I think we've all thought a lot about viral pandemics, and that's kind of what this was all about. If you're interested, a way to turn the clock back a bit is to read this book. You can find it online, I'm sure. Randy Schiltz was a reporter, very early reporter for AIDS in San Francisco, died of AIDS himself, and wrote a journalistic history of the early epidemic that it really is great. And it's a good way to start getting into this. The first report that we had of AIDS was in a journal called the Morbidity and Mortality Weekly Report from the Centers for Disease Control. This came out in June 1981. Strange cases of an unusual infection appearing in gay men in Los Angeles. And we, as physicians, we tend to all look at the MMWR. This has been, and some of you know, in the news more recently with changes at the CDC, but especially important in the early days of the epidemic. This is how we knew some of what was going on. And what we knew is that this was an unusual new disease. And I saw my first patient. This is my first patient on July 1st, 1981, when I started on the faculty of the University of California, San Francisco at San Francisco General Hospital. This was a 22-year-old man with horrible wasting disease. And really, just to make the point, even very early that we knew this was something new. This is a disease that we had never seen before, nothing like this. And at that point, and I'll show you some of the data in just a bit, there was no hope for these patients. So the question is often asked, how did all this happen? How did it spread? And to remind ourselves that it happened quickly, it spread quickly, and we had very few tools to follow this. Unlike the situation with COVID, with SARS-CoV-2, when we knew the virus, we knew the sequence almost immediately. It took us a long time, relatively speaking, for the AIDS epidemic. We now know that this was a zoonotic disease. It was a virus that jumped from chimpanzees, from primates into humans. It spread in Africa where it originated. It followed the transportation systems of the time. It spread across Africa. It was a low-grade epidemic until we began to see cases again here in the 1980s. We now know through very sophisticated genetic sequencing where this virus evolved and how it spread, where it spread, and when it spread. It spread from Africa to the Caribbean first, from there to New York. It probably landed in New York in about 1972. And then it spread to San Francisco in the mid 1970s, late 1970s, and from then on to really to the rest of the world. This was a truly global epidemic, and all pandemics follow the most rapid transportation, and this followed the pathways of jets. HIV was frightening for a lot of reasons, but one of the important reasons is that it was really uniquely dangerous. This slide shows how rapidly AIDS from HIV infection became a lethal and hard to ignore epidemic. It became one of the most common causes of death in young people, especially young men, where the disease was focused. And in contrast to really any other infectious disease, HIV without treatment is uniformly fatal. 100%, almost 100% of people that are infected with HIV without treatment will die of AIDS. This is it's worse than smallpox. It's worse than COVID for sure. And the only disease that rivals it is symptomatic rabies, which is essentially always fatal. So horrible disease with an agent that at first wasn't even understood. And just to say, before I talk about some of the response to the epidemic, that we had very few tools at our disposal. If you turn the clock back, you realize that PCR was only recently available in the early 80s. We didn't have gene sequencing. We didn't have monoclonal antibodies. We didn't have cell sorting. We didn't have personal computers. We didn't have email. And so it was a disease where our response in the scientific community was somewhat slow compared to certainly the way it is now. Communities, especially like those in New York, San Francisco, and Los Angeles that bore the brunt of the early epidemic, were really important in how the disease was approached. And the community response was really important. And I'll talk a little bit about the medical and the response in the community, the gay community especially affected, and very importantly, again, in contrast to some of what we saw with COVID, with the political community as well. The early AIDS epidemic was heavily concentrated. The care of the patients, because people were quite afraid of this disease, and it was affecting a group of people who were stigmatized in other ways, was concentrated in some facilities. And where I happened to work at San Francisco General Hospital, this is a public safety net hospital connected to the University of California, San Francisco, but this is where the epidemic really landed largely in San Francisco. One of the characteristics of the epidemic was a broad response. People came into it from their own perspectives. I was an oncologist. Marcus Conant, who you see here, a dermatologist. We saw the cutaneous, the skin manifestations of the disease first, but other people saw this disease from an infectious disease standpoint because people were getting these unusual and very frightening infectious diseases. And so we formed networks. Again, before email, before personal computers, we talked to each other. We knew who around the rest of the country was working with this disease. And here, my co-developer of the first AIDS clinic was Connie Wapsie, a woman who was very interested in infectious diseases, died herself of breast cancer just as effective therapies became available in the 1990s. But importantly, Connie was very interested early on in how this disease was affecting women, which became a very important issue as we especially saw the epidemic unfolding worldwide. One of the other ways that the response happened in places like San Francisco was the broader community. So in San Francisco, the early epidemic was heavily concentrated in the gay community, and many of the responses took the form of new programs or other programs that were already in existence that changed in response to the epidemic. Ruth Brinker was a woman who was interested in this and created a food program at a time when many people with HIV were really dying of malnutrition. She started a very small program in a local church. Martin Delaney, who was a gay man, but very interested in the community aspects of science, created an organization called Project Inform that sent out a lot of information in the form of printed newsletters at the time. So Project Openham delivering mail, Project Inform delivering information, very key parts of how this epidemic was approached. And as I mentioned, AIDS in politics was an important issue as well. You can appreciate that at the time when most people weren't talking about AIDS, it was key for us, for the country really, to see the public health system respond. And here, our public health director, Merv Silverman, was very knowledgeable and quite responsive. And we had a mayor, Dianne Feinstein, later, of course, a senator, who had become mayor because of the assassination of Harvey Milk and George Moscone. So a political scene that was sort of clued into what was needed in response. Now, I want to talk about kind of the impact at a personal level of the early epidemic in a second. But Michael, why don't you lead people through the next quiz question?
All right. Next question for everyone is, do you know anyone who is HIV infected, has AIDS, or has died with AIDS? Looks like, as everyone can see, 43% said no. Next highest, 27% said 1 to 2 people, and 15% said many people. So at that, I'll turn it back over to you, Paul.
Yeah, thank you. And, I'm going to correct the audience here. I'll bet a lot of people know somebody that has HIV, but isn't aware of it. Because it's now, as Moupali will tell us, such a treatable disease. I was at an event last night and I met a number of people who have been infected for 20 years and they look absolutely normal. So if they didn't tell me they were infected, I wouldn't have known. So it's more common than you might think. So as I said, this spread rapidly. It spread rapidly, and especially in cities like San Francisco, but I'm going to talk a little bit about the fear before we very quickly talk about some of the treatments. So I'm an oncologist. This is a photo of an early patient with Kaposi sarcoma, a cancer that appears on the skin. It actually is throughout the body. But these patients were heavily stigmatized. You can imagine at a time when people were afraid of catching AIDS, that they see a person like this in the community, and they run and hide. People like this were stigmatized even in cities like San Francisco and New York, became unrecognizable because of this disease. And headlines like this, AIDS could end us all. And physicians who in some cases did not respond all that well. Physicians, in this case, a surgeon who was very afraid of catching AIDS, and affected her practice, and a lot of ethical issues that required a response. My wife is a physician as well, and have responded to that. And she and I were having our family at the time. Our kids were born when some of you were being born. Our oldest in 81, our youngest in 87. And this is at a time when I didn't know whether or not I had been infected by this virus. We had no tests until 1985, 1986 to know whether we were already infected. So my nightmare, my recurrent nightmare, was that I had given this virus to my kids. And the fear in the medical community was important. Fear in the public community, especially in the gay community, drove a lot of that anger that was an important part of the activism that was itself a very important part of the response to this epidemic. This slide shows ACT UP, one of the key organizations started in New York, attacking the pharmaceutical industry, attacking the NIH, urging people to do more and faster. And this definitely had a positive effect. The protest led to pressures on drug pricing to make this available. Eventually, the federal government made HIV treatments available to everyone, regardless of insurance. And the attacks also addressed the approval process. Again, something that's important in today's news as well. And the protests led to changes in how the FDA regulated clinical trials to allow them to be streamlined, to drugs can get approved in a preliminary way before they're then licensed. The activism affected us as physicians as well. That's me with hair in the middle, and at a protest meeting march here in San Francisco in 1990. And so we were all involved in this together. Now, I want to go on to talk a little bit about the development of treatments, and again, pass this on to Moupali in just a minute. What about the natural history of disease? I already said that this is a virus that kills everyone. It takes time to do that. This is a slow disease until it's fully evolved. But the importance of how we invested in basic science, basic virology, basic immunology, and especially as we had more of the technologies available to help us understand this better. For example, when we saw our first patients, we had one cell sorter in all of the University of California, San Francisco, one cell sorter. And this is a tool that obviously is now routine. It's in probably hundreds of them at the university. We had drugs beginning in the mid to later 1980s, starting with AZT. This is Zidovudine, was the first drug approved for treatment. But this slide makes the point from one of my clinical trials that it did something. It slowed the disease, but it didn't reverse the disease. So in this trial, people that got treated with the drug compared to placebo died slowly, more slowly, but they still all died. It wasn't until the mid to late 1990s, beginning in 1994, but then especially in 1996, that the concept of combination therapy, the idea, as we do with cancer, as we do with infections like tuberculosis, that combination therapy using multiple drugs that hit the virus in different places might be more effective And with the development of protease inhibitors, the first of these potent new classes of drugs, in combination with the older drugs like AZT, we began to see the real effect. And this slide from the New England Journal shows that as these drugs came into use, the mortality rate for AIDS dropped precipitously. And again, to this point where people are living now very long lives without treatment. There's a gay newspaper in San Francisco, the Bay Area Reporter, that used to have small little obituaries, tens, 20s, hundreds of them in each issue. And they thought that it was significant enough in 1998 for the first time the issue had no obituaries. So a long way that we'd come. And the first treatments were successful to some degree, but very cumbersome. People had to take literally handfuls of meds several times a day. But given the pressure and the cooperation between the academic and the pharmaceutical industry, we were able to develop drugs that were much more convenient and much less toxic than the early treatments that we had, so that by the time more recent developments happened, we had treatments where we are now in the form of literally one pill once a day with multiple agents contained within the pill. So dramatic improvements and definitely reflected in the statistics on this epidemic nationwide. We've learned a lot of lessons. I'm not going to go through all of them now, but you can imagine that we still think about this a lot, especially given where we were with COVID. So I want to end with a thanks to certainly to the audience, to the organizations that put this together. Again, great to do this on World AIDS Day, and refer you to a 30-minute documentary that I made a number of years ago called Life Before the Lifeboat. It's on YouTube. I think we'll give you the link to that, but it goes through some of this. And again, you can see some of this in our earlier days. So thank you very much for your attendance.
Hello, good morning and good afternoon, everyone. I just wanted to say it's an incredible honor to be invited to speak today on World AIDS Day and to follow Dr. Volberding, who was a mentor and a person I learned a great deal from when I was doing my HIV training at the San Francisco VA and at San Francisco General. So it's a great honor to be here. I'm going to be talking about some developments in HIV treatment and prevention, kind of taking off from where Dr. Volberding left off. So let's continue. So just wanted to start with drug development for HIV treatment and prevention, having come a tremendous way since HIV was first discovered or first came into being in our awareness in 1981. And my institution that I work for in Gilead was founded in 1987. And it highlights our HIV drugs. So we started with developing some NRTIs initially, and then we helped develop our first single tablet regimen, helping going from that handful of pills you saw in Paul's picture to one tablet once a day, which was a huge advance in 2006. And then subsequently, one of the things that we've worked on is improving that single tablet regimen and developing stronger and more potent and better versions of the earlier medications to improve tolerability, efficacy, and people's quality of life on the medications. The other huge breakthrough is pre-exposure prophylaxis, which is HIV prevention. So just like when you travel to, let's say, a malaria endemic location and you take medicine to make sure you don't get malaria, that's called pre-exposure prophylaxis. You're taking it before you're exposed to the infectious disease in question. Similarly, for HIV, we can do pre-exposure prophylaxis, which people since 2012 have been able to take a Truvada, which is tenofovir disoproxil fumarate, um, um, combined with tricytidine, and that is FTC. So that's been available since 2012. And then we also developed Descovy, which includes a different updated form of tenofovir as for HIV prevention, which was approved in 2019. But those are daily pills. And a lot of times, as we have lived through this era, we've seen that not everyone is truly benefiting from an HIV treatment or prevention regimen. And to bring more people into care for treatment and help them become virologically suppressed, which improves their own health and quality of life and also decreases to zero if they're undetectable, the ability of the virus to transmit to another person, and to help people prevent HIV if they are HIV negative and do not want to acquire HIV. We have been innovating on other options for HIV treatment and prevention, including trying to develop long-acting regimens. So before I get started, I do want to say that as we innovate, we need to keep something in mind. And one of my mentors in undergrad at Harvard, with whom I worked later on, Dr. Paul Farmer, who founded Partners in Health. This is a picture I took of him when I was doing a rotation with him in the year 2000, wrote a book called Infections and Inequalities. And in this book, he had one of my favorite quotes, which is, infections track along the fault lines of social inequality. And I would say my corollary after my experiences in working in drug development is that innovation without intention can unwittingly exacerbate these disparities. So it's really, really, really critical that we are intentional in our innovation. And as we look for new options, whether it's for HIV treatment or prevention, but specifically in HIV prevention, I think there is a critical need to innovate both in the science and in health equity. One without the other can unwittingly exacerbate this inequality. And in the program I'm going to be telling you about, we focused on both of those two important twin priorities of innovating in the science and innovating in health equity. So lenacapavir is the drug that I'm going to be talking about. It is a small molecule that's highly potent, and it's a capsid inhibitor. And for people who are unfamiliar with the HIV viral cycle, here's HIV, here's the nuclear material. It enters through the cell and goes into the nucleus so it can hijack this is a human cell and this is the virus. So it can hijack our nuclear machinery and make copies of itself and then make new viral particles. And then HIV can leave the cell in a new viral particle and go on to infect other cells. Now, capsid inhibitors work at multiple of these steps that I will show you. And lenacapavir is a drug that has been approved for HIV treatment and prevention at this point. For treatment, it's approved as part of combination therapy with other drugs. And for prevention, it has been approved by showing efficacy and safety as a single agent. And I just want to attract your attention to this green structure, which is the capsid. And lenacapavir is a drug that works by inhibiting the proper development of the capsid and affecting its function at all these steps. So we are focused on going from our current options in treatment and prevention to breakthroughs and hopefully ultimately game changers. Okay, so now for a chemistry question, and I will turn it over to Michael.
All right. And the question for everyone is how many pentagons are in a closed polyhedral fullerene? Would that be 4, 8, 12, or 20? And we almost got to no one voting for 4, but at the end, a few people did. The highest was 47% of respondents said 12, and 29% said 20 for the 2 highest. That'll turn it back over to you.
All right. Strong work to the 47%. So HIV capsid is a fullerene cone. So it's a subset of fullerene polygons, if you will, basically, you may have heard of P24, which is an antigen from HIV that we use when we do 4th-generation HIV testing. So we'll look for P24 particles in people's blood, and that lets us know if somebody has been acutely affected with HIV even before our antibodies come up and we start making our antibodies, which is the HIV antibody test. There's an antigen test, and that's P24. And so capsid is a monomer that is part of this gag protein, and it is at P24. Now, it's cleaved by a protease, and it makes 1,500 copies of itself, and that makes this beautiful polyhedral structure that is the capsid core, and it contains 250 hexamers and 12 pentamers. And this is a mathematical fact. I'm not a mathematician, but I know that a lot of chemists are familiar with the fact that Buckminster fullerene or a soccer ball also has exactly 12 pentagons. And that is because that's the way that this structure comes together. And there are probably people on this call who can explain it better than me, but I just thought it was really interesting that there's always 12 pentamers in these fullerene structures, and capsid specifically is a fullerene cone. So for people who are in drug development, they understand how hard this is. And I just want to show you all the things that our brilliant chemists and scientists try, all the different molecules. So I'm going to play a little video, and let's hope it works. And you can see the pocket where Lenacapavir fits, and all the different drugs that we tried, all the different tests, all the different side chains that we added to get to the molecule with the highest potency and tolerability and the properties that we were looking for. So people are very familiar with the design, make, test cycle. And this is not just innovation in chemistry through iterative innovation, but I'm trying to go to the next slide, but it also requires innovation across formulations and process chemistry, because a lot of the formulation innovation comes from not only the chemistry of the molecule, but the excipient and how we put the molecule together for a final product. And the capsid project started in 2006. And this target was considered undruggable because it was a protein target and it required innovating in protein folding and protein aggregation. And our first thought was to try to inhibit the way the capsid came together. And that was our first lead, but it didn't end up working. So there was a very brilliant scientist at the company who came up with the idea, what if we, instead of trying to inhibit how capsid formed, what if we make capsid, the structure, form more quickly and actually have an accelerator instead of an inhibitor of the process? Because he surmised that perhaps that would cause defective particles, and he was right. So that's the 2nd-generation lead that we've been working on since 2010, but we've been working on capsids since 2006. And then between 2010 and 2016, it took us each dot is one of the molecules that we tried to get to our most desirable clinical candidate in 2006. And then we first developed it for highly treatment experienced people. So people that Paul may have treated from the beginning of the epidemic who had multi-drug resistant HIV from being on serial monotherapy and trying different formulations and challenges with those therapies. they needed new options, and adding lenacapavir to an optimized background regimen was able to durably virologically suppress their multi-drug resistant virus. And so that drug was approved as Sunlenca in 2022. And we started studying lenacapavir for HIV prevention in 2021. And since then, we'll tell you the results in a second, but the molecule has been recognized by various organizations and entities for its innovation and breakthrough, because it can be given as a twice yearly subcutaneous regimen. So it's quite long acting and reduces the burden and challenge of daily adherence to a single tablet regimen. Back in the late 90s, we thought that a single tablet regimen was the holy grail, and that was so amazing to get from a handful of pills to one pill. But now for HIV prevention, instead of having to take a daily pill, you can get injections twice a year with very high efficacy and safety. So the PURPOSE program, which developed lenacapavir for HIV prevention, had two pivotal trials. PURPOSE 1, which took place in cisgender women in South Africa and Uganda, and PURPOSE 2, which took place in cisgender gay men and other men who have sex with men, trans women, trans men, and gender non-binary people across several continents in the United States, South Africa, Latin America, and Peru, Brazil, Mexico, and Argentina, and also in Asia and Thailand. And all of this work happened on the foundation or the roots of the innovation in the molecule that was evaluated for safety and efficacy in the treatment program, and then also with a focus on partnerships, including the voice of the people most affected by HIV who would benefit from PrEP and the community advisory groups who represent them, a focus on person-centric design with an eye towards person-centric PrEP for when the molecule would be available for use, and a real focus on making sure that we included those people who are most disproportionately affected by HIV into our trials to make sure that we were, again, not unwittingly exacerbating the disparities by making sure that we were including the people who are most disproportionately affected and really focusing on inclusion, diversity, and equity. Of course, we followed the ACT UP. Paul mentioned the activists and what they championed in the 80s and 90s and how they improved the drug development process. And they really talked about nothing about us without us. And we championed that. So we started our first community advisory meeting with civil society, government stakeholders, scientists, clinical investigators, their staff, and our team members to ask folks about some of the innovations we were going to follow in the PURPOSE program, or we intended to, such as including pregnant persons and including adolescents in adult trials to reduce the time it takes to have data on those populations by including them in the first pivotal trials done. And there was a woman at this meeting. So historically, pregnant people haven't been included in pivotal clinical trials because people have been concerned about untoward effects on the fetus, which is a reasonable concern. But there are ways to safely include pregnant people in clinical trials, and we wanted to follow all of those steps and do that because we know that pregnant people are disproportionately affected by HIV, particularly in sub-Saharan Africa, and adolescents are also disproportionately affected by HIV globally. And so we wanted to include those two populations, but there were a lot of ethical and regulatory concerns that were raised, and people were a little reluctant to do something different. But one thing that was critical that happened was we had invited a woman who hadn't been obvious to me because we were at a conference table, but she stood up and she was quite pregnant. And she said, I'm a 17-year-old person and I am pregnant and I want people like me and my friends to be included in prevention trials because we need new options and we don't want to be left out. And I don't want to get HIV and I don't want my baby to get HIV. And her personal story was so compelling that along with all the data and the safeguards and the preclinical information, it allowed us to truly change hearts and minds and do something different, which is including people who are pregnant who to continue in the clinical trial on study products so we could understand the efficacy and safety and clinical pharmacology of the product in pregnant persons. So this is just a highlight of our committed and consistent community and stakeholder engagement for Purpose 1 as well as for Purpose 2. We met every other month with our established community advisory groups, and this really helped us to ensure that we were addressing those 4 critical pillars of purpose to make sure that we were innovating with intention. These are some of the things that people asked for, and we were able to deliver on all of these requests given to us by the community advisory groups and other key stakeholders. So we're very delighted to share that this was the most globally, racially, ethnically, and gender diverse phase 3 prevention and likely treatment program conducted to date. Let's go back. I advanced too quickly. I just wanted to highlight the countries, and the percentages are which countries contributed the most populations to these 2 programs. And in Purpose 2, we're really focused on including those groups who were disproportionately affected by HIV globally and in their countries. So we were delighted to see 67% people of color, 63% people of Hispanic and Latin ethnicity overall, and 22% gender diverse, because these groups are disproportionately affected by HIV. So we saw unprecedented efficacy in these 2 pivotal trials. There's never been a PrEP trial with 100% efficacy, and those were the results of Purpose 1. 0 cisgender women or adolescent girls acquired HIV in Purpose 1. In Purpose 2, the study of cisgender men and gender diverse individuals, 99.9% of people in the Lenacapavir group did not acquire HIV. So really high efficacy compared to prior PrEP trials, which did not show this high efficacy. Again, we talked about the inclusion and how important it was to include pregnant people in Purpose 1, and the available pregnancy outcomes were similar to those expected for the population. So there was no increase in higher rates of interrupted pregnancies or, for example, spontaneous miscarriages. One of the other stories I want to tell is that on the upper left, you see a picture of where participants were coming. They were in Uganda, and where they were delivering across the top row of photos. So this is the maternity ward or maternity wing for delivery. And one of the challenges for a lot of our participants is, in order to be seen by medical staff in underserved locations, the onus is on the pregnant women to bring with her supplies, including gloves and other materials to help the care staff care for her during her delivery. And a lot of people didn't have the resources to bring these supplies with them from the rural areas where folks were coming for delivery in Uganda. And so we worked with the sites and the investigators and internally with our ethical and legal and regulatory consultants to come to a solution where we could provide a mama kit with these resources. And this allowed us to have this data in pregnancy and in breast milk and infants, and really supported the safe care of pregnant people in our clinical trials. And on the right-hand bottom right is Purpose Mamas and Babies from the Uganda site. And I want to highlight that these the participants gave full consent for us using this photo. And I think this is really critically important because a lot of times, especially in the West, we have very, very strict rules on undue enticement. So anything that could be seen as an additional value has to be taken into consideration as part of the participants' reimbursement and support that they're receiving through the study for participating in the study, and that there's really, really, I think, an appropriate allergic reaction to giving people anything that could be seen as a financial benefit for being in the study beyond other folks. And this comes from a great place because we don't want people to feel obligated to participate and receive extra benefits by participating. But at the same time, that consideration needs to be balanced with appropriate, safe care of pregnant people in clinical trials. And for the Uganda sites, providing this mama kit really helped make a difference. And telling the stories of where the women were coming from and where they were delivering helped us, again, change hearts and minds of folks to support this extra support for pregnant people in Purpose 2 in the Uganda sites. So just to review briefly, Purpose 1 intentionally included pregnant and lactating people in our phase 3 clinical trials, and both Purpose 1 and 2 intentionally included adolescents. With respect to Purpose 2, we focused on gay men who have sex with men and trans women of color because they are truly intersectionally disproportionately affected by HIV. But our trial was the first to intentionally include trans men and gender non-binary people. And again, we included adolescents with a focus on recruiting young people in Purpose 2. So now, after we've gotten the results of the trials, we now have everybody participating in what we call OLE, or an open label extension, and 95% of people voted with their feet to enter the open label extension. As I mentioned, Lenacapavir is a twice yearly subcutaneous injection, and in the primary part of the study, we studied it only in the abdomen. But now that we're in the open label extension, participants can be receiving it in the upper arms, their upper buttocks, and in their thighs as subcutaneous injections as well. And again, 95% of people chose to either continue Len or start Len if they had been randomized to the oral prep arms in the clinical trial. There are many firsts in both our focus on innovation in the science and innovation in health equity. And I don't want to read all of these to you today, but I just want to highlight that there are several unique aspects of the science, including a counterfactual trial design, which is another lecture altogether, our innovative twice yearly product, as well as nested PK studies, novel ways of recruiting and retaining folks, and this focus on person-centric design, as I mentioned, with pregnancy and special innovations for including adolescents. And this all happened in an ecosystem of partnership with really robust engagement of the investigators, the community advisory groups, setting specific recruitment plans and inclusion goals, and tailored participant engagement based on where they were and who we were trying to recruit, and also collecting quantitative and qualitative data about how people felt about the different products in the clinical trial. And we think all of these efforts together led to not only the unprecedented efficacy observed, but also the distribution of people that we were able to include in this trial. Okay, I'm going to turn it back over to Michael for our last question.
And our final question for all of you is, on average, how many years after adult data is available do pregnancy or pediatric adolescent data appear on antiretroviral drug labels? Your choices are 2, 5, 7, or 10. It looks like the highest percentage of respondents, 38%, thought it was 5 years. Next highest thought it was 7 years with 28%. And coming in third was 10 years at 18%. So that'll turn it back over to you.
All right. Thank you, Michael. So it's kind of a trick question because the number is changing throughout the epidemic, but the number that we normally quote is that about a 10-year delay to get pregnancy, lactation, infant, pediatric PK, or adolescent data into clinical trial labels. And because of the aspects of the program that I described, our intention on inclusion, we were able to not only have this very robust, broad indication, but also, information on pregnancy, lactation, and adolescence to support folks. The other thing I want to talk about is regulatory approval alone is not sufficient to get Lenacapavir to the people who need it the most. Not only do we need innovation in the science and the health equity in clinical trial research, but we also need innovation in access. So we pursued multiple attributes to ensure that we were setting ourselves up to make sure Len got to where it needed to go globally. And I'll just highlight our global regulatory filing strategy. So we filed in the United States and in Europe. And I just want to say that usually most companies and sponsors file in the United States and Europe, and there's quite a delay to getting drugs approved in sub-Saharan Africa. But in this case, we had our first approvals in the same year, with less than half a year after the approval in the United States and in Europe. And this is really due to some innovative approaches that we took, including the WHO collaborative registration procedure. So once we got the positive opinion from the regulatory group in Europe, the CHMP, that led to the beginning of the WHO pre-qual process, which was received in October 2025, which meant that we could start filing in our collaborative registration procedure countries and these other countries. And globally, the team has been incredibly busy. So highlighted in dark gray are all the places where you've applied, and highlighted in blue are the 3 countries that were approved when I made the slide. And we just heard today that Malawi has also been approved. And the team is working very hard to finish the rest of the approvals in the CRP countries, as well as other critical countries by the end of this year, the beginning of next year. And I'll just start closing by talking about all of these different ways that we're making sure the drug is getting to places. So we transferred our IP and signed voluntary licensing agreements with generic companies to make generic versions of Lenacapavir even before we filed with the FDA or received approvals anywhere. And we also worked very closely with the U.S. State Department and the Global Fund and various other companies and institutions globally to make sure that we were getting Lenacapavir before generics are able to make enough supply. We expect that that will be coming in 2027, 28 across the globe. We're supplying enough Lenacapavir for 2 million people over 3 years at no cost, because we know it takes about 3 years for these 6 generic companies to get their supply up and running. So we're very excited that in November 18th, we were able to get the first supply to Eswatini, and you can see it's starting off at our processing facility in manufacturing facility at Gilead, and then getting to South Africa and Eswatini and all of these places. And this was an event with the G20 that took place in Eswatini with supply of Lenacapavir reaching there. And the other thing I wanted to highlight here is I had the opportunity to see Lenacapavir administered in a mobile van in New Haven by Dr. Sandy Springer and her team to a person who injects drugs as a PrEP option for commercial supply in the United States. So pretty much getting to hard to access folks in the United States and globally to the places with the highest burden of HIV. So the other thing I want to announce is that we have also are moving from purpose to propel to focus on implementation science that will be increasing access and uptake of Lenacapavir. And the 2 Unitate projects in South Africa at WITS RHI in Johannesburg and ImPrepLen with Dr. Grinstein in Rio have started enrolling, and Lenacapavir is being given in these phase 3B studies, implementation science studies, to understand critical questions about uptake and barriers and facilitators to Lenacapavir use for PrEP. And we are not stopping there. We've also now developed a once yearly version of Lenacapavir, which would be a true game changer. And that has started enrolling. So here, our purpose tree is becoming a purpose forest with the once yearly version, our implementation science studies in the United States and globally in high burden regions. And I'm delighted to conclude by just thanking all of you for your attention and thanking the ACS and the Science History Institute for the opportunity to present. And if you want to know more about our access strategies, you can scan this QR code, and there's the link below on our access page, which is updated more rapidly than my slides, which are already out of date as of like 6 a.m. this morning. And we're really focusing on this triple approach of innovation in the science and health equity so we can innovate in access and try to truly end the epidemic for everyone, everywhere. And I just want to thank particularly the participants, their families, their friends and community in the clinical trials, as well as our site staff, community advisors and investigators, and all the folks internally who made these trials possible. And here's my contact information, and I will take down the slides and turn back over to the team.
Thank you both for excellent talk. We have some good questions coming in, and I think I will give the first question to Dr. Volberding. Yes, you talked about the capillary sarcoma, and some of the audiences are curious, what's the relationship between cancer and HIV?
Oh, yeah, it's a really great question. And it's not an easy one. There are some cancers like capillary sarcoma that we see a lot in HIV. We see lymphomas as well, non-Hodgkin's lymphomas. Most of the cancers that we see are not so directly related to the immune system, but we think that that HIV, the virus, as it destroys the immune system, damages immune surveillance. So cancers that otherwise happen at a low rate happen at a much higher rate. So the answer is we see kind of a spectrum of cancers, but some like capices and lymphomas at a very high rate.
The second question will go to Dr. Deck. So it's very impressive that you're talking about Lenacapavir only needs to be taken twice a year. So instead of, one pill a day for every day. So what do you think is the mechanism? Like, why is it so long-lasting?
Great. So this is really important to understand, and I didn't get into it, so apologies for that. But it's really a property of the molecule. It's incredibly highly potent, and it's relatively insoluble. So it lives under the skin as a depot. It's not the excipient or necessarily the formulation that makes lenacapavir long-acting. The excipient and the formulation allow better properties of the final product, but it's really lenacapavir's unique chemistry and nature that makes it insoluble and highly potent at small doses, which allows us to deposit a certain amount of it under your skin and have it elute out and enter your bloodstream for over a 6-month period and give protection at the right levels for over a 6-month period. I hope that's helpful.
Yeah, definitely. Thank you for the answer. Dr. Volberding, your question about asking people whether they know people who are infected with HIV, I think definitely about some people wonder, they don't even know what are the common symptoms that may appear during the acute phase of HIV infection. Can you elaborate on it?
Yeah, spectacular question. So the symptoms of acute HIV infection, i.e. kind of when somebody first becomes infected, look a lot like the flu, fever, just fatigue. But those go away after a couple weeks, and people feel absolutely normal. In many cases, they don't remember even that they had those symptoms. Then the virus sits in their immune system and gradually kills it, basically eats away at the immune system. And only after a number of years, 5 or 10 years, does the immune system become so damaged that people then develop symptoms either the wasting that I showed in that one slide, where, as with cancer, people lose weight, they can't maintain weight, or they develop cancers or other infections. That's when they became really ill. And that's when every one of our patients died. In the early epidemic, we tended to see people as their immune systems were already being damaged, and 100% of those people died from the disease until we had the kind of treatments that Moupali talked about.
Thank you for that. That was definitely a devastating debate.
Yeah.
And quite an achievement right now would make it a livable disease
Yep.
and in the future preventable.
Yep.
So that's fantastic. So for Dr. Das, we wanted to know, for this wonderful preventative drug, is there any potential side effects that you wanted to?
Yeah. So the most common adverse events observed in the clinical trials, we could bucket them in two groups. One is, some nausea, GI intolerability, that would be in like one group, headache. And then another group of most commonly reported adverse events, because anything that happens to a person in a clinical trial gets reported, is sexually transmitted infections. In both Purpose 1 and Purpose 2, there was a really high burden of STIs. We diagnosed them and treated them, but we did asymptomatic screening and picked up a lot of sexually transmitted infections. Separately, related not to what was happening with the people that could be potentially attributed to the drug or to the epidemiology and background burden of disease, there was also attributes related to the fact that it's an injectable. So any injectable drug is going to be associated with some amount of pain or erythema or swelling at the site of injection. And indeed, those are the most common side effects from the initial injection. Now, as I explained briefly, not during the presentation, but during my answers, that lenacapavir forms a drug depot that lives under the skin. So in the fat layer between the skin and the muscle, or what we call the subcutaneous layer. So there are a lot of people who were able to feel but not see that drug depot. And that ability to feel the drug depot depends on how much sub-Q fat you have. So it's kind of like the princess and the pea. So if you don't have a lot of sub-Q fat and you run your finger over the area, you may be able to feel the nodule, which is the drug and the body's response to the drug. And if you do have sub-Q fat in your belly or in some of these other sites where, like basically the flanks or your love handle, and you get the injection there, it might be harder to feel or appreciate the injection. So those are the most common side effects from the injectable. There are no renal or liver or other lab abnormalities noted. In fact, the renal function was improved in the lenacapavir arms as compared to the Truvada or Descovy arms, because there was no tenofovir component, which does have an impact on renal function. A small impact in a small number of people, but we were doing thousands of people. So that impact obviously was not seen in the lenacapavir arms.
Great. That's excellent. Thank you both, Arnold. Dr. Volberding, I think that there's a question for you. Talking more historically, to the West African origin place of the HIV. So African voices is largely absent from the HIV prevention and cure. So can you comment on how they manage it more and how it is right now?
Yeah. So Moupali introduced the idea that epidemic diseases kind of identify problems in the social structure that we have. And we saw that clearly with the AIDS epidemic, where it started in Africa, but it wasn't appreciated. It was just seen as yet another infection in a part of the world with a lot of other infections. And it wasn't until it hit the U.S., frankly, that we paid attention to it. And even as we developed treatments here, even though people were dying by the thousands in Africa, there was no attempt to make those treatments available in Africa, especially in East Africa, Uganda, especially where the epidemic was initially really focused. So it reveals kind of how we were willing to ignore or just refuse to deal with the reality of this epidemic. I'd have to say, though, that HIV, the AIDS epidemic in Africa showed us that when we decide to do something, we can do it. So treatments were made available widely through the PEPFAR program in the U.S. Agency for International Development at very, very low cost. So the best drugs that we had here in the U.S. Became available and to some degree still are available in even the poorest parts of Africa. So great. The problem that we have now, of course, is that, again, AIDS reveals the social problems that the U.S. has decided to pull back those programs so people will now not be able to get their HIV drugs. And so predictably, as I said, everyone without treatment will die. We will see people dying unnecessarily of this disease, a huge challenge to us.
Thank you for that. Definitely there's some more physical viral as well. Dr. Das, since antiretroviral mechanism of action, can you comment on whether you can see it being used in leveraging other retroviral diseases for their diseases as well?
Lenacapavir specifically accelerates the development of the capsid, which is unique to HIV. So it is specific to HIV. And as far as I'm aware, does not affect other viruses. I can follow up and get back to you. I don't think it's been studied in anything else because it's so specific to HIV capsid.
I have two questions that are for both of you. One is, what lessons can we adapt or learn from the community dealing with HIV and AIDS to our current resurgence of infectious diseases, right? Of non-go COVID and still is around you back. So, any comments on both our analysts?
Sure, I'm sure we both do have thoughts. I'll start. The community response, the broadly defined community response to the HIV epidemic was absolutely essential. And it provided services to people, it helped decrease the stigmatization, and it made the best treatments available worldwide. And it allowed substantial investment in the basic science, which proved to be absolutely critical to developing the treatments and the preventions that we now have. I think that the one of the big issues, there are a lot of them, but one of the big issues is the importance of a public health infrastructure. So if you ask the question, how many people are getting infected? How many people are living with HIV? How many people are dying with HIV? Those are all questions that need data. And the U.S. has a pretty good public health system, not nearly as good as many countries, but the ability to collect that data and understand it at a national level is really important if we're going to be responding, if we're going to pick up the next COVID pandemic. And that's where I think most of us are really concerned that we seem to be moving away from support for the kind of public health infrastructure that we know we're going to need. We know where there'll be another epidemic. So it's just a question of when.
It's an ecosystem of partnership and care, and we need absolutely, the community played a huge role and continues to play a huge role. And there has been a reinvigoration of community efforts as appropriate. There's it's like a sine curve, goes up and down with respect to what's needed. And there's been a robust re-engagement of community as required. And then in a lot of places, community has continued to lead since the beginning in global locations and really advocating for more access. That's been a consistent theme. And I think we need strong institutions in academic medicine and, translational research, basic science. We need strong public health institutions locally, state, federal, global. And we need strong support for innovation in biotech and industry to help develop and all of the help develop new options and make sure the options are getting where they need to go. And all of these groups need to interact and push and pull on each other. And the advocates do a great job of making sure and holding folks accountable to what their role is in this ecosystem of helping people have better, healthier lives, which I think we're all involved in some aspect.
Yeah, absolutely. The complicated question is a complicated solution. So on the last question, we have a very young student here, eager to get into the field. And I wanted to pick both your brains to tell him or her what is the most critical skill that they need to achieve in order to work in this field.
I'll start. I think that a desire to respond to the need that you see around you to people with these kind of problems. I think that it depends. Some people are coming into this with a public health perspective. Some people are educators, and that's really important. Some people are scientists. So I guess the answer is it depends, but it's all driven by your interest in helping other people.
I totally agree with Paul. And I think the only thing I would add to that is a piece of nerdy, practical or pragmatic advice, which is make sure your LinkedIn is up to date. And if you're applying for jobs, read the job description and the announcement very carefully and make sure that you have the keywords in your profile and in what you're doing, because lots of places are using AI to screen and you want to get past that initial screening. So make sure, just like if you're writing a grant, you're responding to the grant announcement, you have to make sure you're really responding and tailoring for the jobs that you're most interested in. So that's my pragmatic advice.
Excellent.
I love that. That's a complimentary answer to the question. And thank you both for the wonderful talk and for the wonderful Q&A. So now I'll turn it back to Grace and you all have some more announcements.
Thank you. Thank you, everyone, for that fascinating discussion. It really takes me back, particularly thinking about no email and 100% mortality. As we conclude today's presentation, I'd like to remind our audience that you can find past Joseph Priestley Society programs, including our last month's talk on the new horizons in transplantation, on our YouTube channel. More stories about HIV can also be found at the Science History Institute's website. We have a discussion of AIDS activism in an episode of our Distillations podcast about patient-led advocacy efforts. We have a Distillations interview with historian Gregory Morgan talking about the development of a test to diagnose HIV. We have an oral history with the physician Vishva Dixit describing what it was like to encounter patients with HIV before there was knowledge of the virus. Finally, just like our talk today, the very best part about the Science History Institute is that access to our articles, collections, podcasts, and events is free. Your gifts power our programming. Finally, we are already planning for our next ACS JPS collaboration. It's going to be in February 6th at 1:00, and it's themed for Valentine's Day, chocolate, roses, wine, the chemistry of romance, flavor, and drink. We hope that you'll be joining us. We'll be sending information about that fairly shortly. Thank you very much, and thank you for joining us today.
Thank you for watching this presentation. ACS Webinars is provided as a service by the American Chemical Society as your professional source for live weekly discussions and presentations that connect you with subject matter experts and global thought leaders concerning today's relevant professional issues in the chemical sciences, management, and business.